but I actually kind of lump it in with the lipid. It's a little bit like we did with enzymes, right? We're going through the biological molecules. We talk about amino acids and proteins. Well, as long as we're talking about proteins, we might as well get in all aspects of enzymology. And so this is a little bit, as long as we're talking about lipids, we might as well go ahead and use that to start talking about membranes and membrane functions. So we will, so we're doing that, and so yeah, so it's basically through that, what we get through, I'm hoping to get through it all today, doing that and all the other carbohydrate and lipid stuff, that's what's gonna be on the midterm on Tuesday. So it's sections 4 and 5. But I'm going to continue this kind of with the same outline. Roman number 1 was let's see. Roman number 1 was the structural types. Oh no, actually this is number 2. Roman number 2. Number 1 was the structural types. Roman number 2 is going to be membrane structure and function. So, you know, a lot of times when I draw a membrane, I'll do that little thing, you know, this thing like that, and put a whole bunch of them like together, and you see a lot of depictions of membranes like that. But that's like gross oversimplification. So what we're really going to try to do is get into a little bit of the complexity about what a membrane really is. And so the first thing is we'll look at composition. And one of the first things we want to say is that if we look at, like, what do you think is the major component of membranes? You know, most people think, okay, well, it's the phospholipids, right? That's the major component. But actually, by weight, there's more protein in a membrane than there is phospholipids. So, so by weight, protein, and this is going to depend upon, this is going to be a point I'll be making over, membranes vary quite a bit from organism to organism, from cell type to cell type, and then from membrane type within a cell. But generally, protein is 45 to 75% by weight. Lipids, 25 to 55%. And there's even some carbohydrates. I think a lot of the carbohydrates are actually attached to the proteins. The carbohydrates, 0 to 8%. So the point here is that, you know, the simplistic view of a membrane is a phospholipid bilayer, but the reality is it's a phospholipid bilayer with a whole bunch of proteins embedded in it. And some of those proteins have sugars attached to it as well. The other aspect, point I basically want to make is that the composition is characteristic for different membranes. So what I mean by that is that a plasma membrane has a different composition in terms of proteins, lipids, carbohydrates. And what we'll see is even the different types of lipids, the type of proteins, the types of carbohydrates is going to be different in a plasma membrane then from as a Golgi apparatuses, then from an endoplasmic reticulum, then from a nuclear membrane, from a vacuolar membrane. Also that the plasma membrane of an intestinal cell is going to be different than the plasma membrane of an erythrocyte, is going to be different than the plasma membrane of a nerve cell, right? There's a lot of variability in here, and we're going to get to a little bit more of the details of that. The other thing I'll say is the fluidity is controlled by fatty acid composition and cholesterol content. And cells will actually change the fatty acid composition and the cholesterol content depending upon their environment, and particularly with temperature. You know, I said the higher the temperature, the more fluid a membrane tends to get. And so at a higher temperature, because it basically wants to maintain a certain level of fluidity, so at higher temperatures it's going to have more saturated fatty acids and more cholesterol. And at lower temperatures, it's going to have more unsaturated fatty acids and less cholesterol in there. So, okay, so now I want to get into, this is sort of some aspects of the overall composition. Now I want to get in and talk about some specific ways lipids will vary in different cells. So this is lipid heterogeneity. First of all, we'll talk about asymmetry across a membrane. That if we have the membrane here, I'm going to draw this, overly simplified version of a membrane again here. See, this is a plasma membrane, and this is the outside here of the cell, and this is the inside here. That the composition of the phospholipids on the outer layer are going to be different than the composition of the phospholipids on the inner layer.
Is that what heterogeneity?
Heterogeneity, yes. So I have an example here, an erythrocyte cell membrane. The outside tends to have high phosphatidylcholine in there, high sphingomyelin levels, and the inside tends to have high phosphatidylethanolamine levels and phosphatidylserine. So again, the specific the specific phospholipids that are on this outer side are going to be different than on the inside. So then that becomes a question, well, how does that happen, right? You would think that it would just kind of all equilibrate, and that's tightly controlled, and there's a series of enzymes. So if we look at movement of lipids, across the membrane is enzyme controlled. I mean, it's not real easy to, if you have a polar head group, there's a pretty high energetic barrier to it flipping across the polar, but it will happen at a certain rate. So if you start out with a membrane that has a different composition on the outside than the inside, over time it's going to equilibrate across there. But there are enzymes that are actually actively working to maintain this different thing. They have things that are called flipases. Flipases actually don't require ATP, and they will just take advantage of an existing gradient, and phospholipid translocases which use ATP.
Professor, when you say across the membrane, are you just talking about them flipping sides,
Right,
whether they're
right,
inside or
yes,
outside?
Yes, exactly.
Okay.
Flipping like back and forth, right? And so there are enzymes that will take basically, if you have a whole bunch of phosphatidylcholine down here, not very much over here, some will take advantage of that gradient, the flipases, and will just lower the energy barrier of getting that polar head group through this hydrophobic core and just flip it over. And there's other ones that will help generate that gradient by actually using the energy of ATP to actually move it against the concentration gradient. Nothing is simple in these biological systems. Okay, so that's a symmetry across a membrane. What about lateral asymmetry? So this is like you have a plasma membrane on the outside of the cell. Well, maybe on one side of the cell it's different than the other side of the cell. Let me give you an example. See, we have an intestinal epithelial cell. And so maybe here is the lumen of the intestine. So here's lungs coming down this way, right? And then you've got other cells here. You know, and the general idea is that the epithelial cells are going to absorb nutrients, and it's going to, sometimes it transforms them, and then it's going to move them to these other cells, and eventually it makes its way to the bloodstream, right? So this part here is known as the basolateral domain. While this part here that's focusing, that faces the lumen is known as the apical domain. Well, these two membranes, in spite of the fact that they're both cell membranes, have quite different functions, right? This function is to absorb the nutrients. This one is to actually export it. And so the lipid composition and protein composition is going to vary quite a bit between the apical domain and the basolateral domain.
Is this one cell that we're looking at?
This is one cell, yeah.
Okay.
You know, there's going to be more around here. And the point, again, is that even if we're talking about a plasma membrane, it's not like there's just one plasma membrane for one cell type. It depends, okay, what are we talking about in the plasma membrane in the cell? So the other place we see lateral asymmetry, actually probably have done something like this, maybe put a little A here, something like this. There are also like microdomains. in which, maybe we have our phospholipid bilayer here. And then all of a sudden there might be an area maybe particularly enriched in cholesterol. Might have a different phospholipid composition. Maybe have sphingolipids instead of glycerophospholipids, and then it'll go back to the way it was. So these little areas here, I'm going to show you a little bit better picture, are known as a lipid raft. So small areas where you have different compositions. And there might be different proteins associated with this that have different functions. So it's not just about lateral heterogeneity across major parts of the membrane, but also heterogeneity more at a micro scale. All right, so this is a model of a membrane that maybe is a little bit more indicative of the overall compositional complexity of the membrane. We do have the phospholipid bilayer, but within that phospholipid bilayer are a whole bunch of different proteins. And we're just about ready to talk about different ways proteins are associated with that layer. But then there's also going to be some carbohydrates attached to these as well. So we have our protein is by weight is the biggest component. Then are phospholipids and a little bit of carbohydrates. Here's basically another. We're not getting into the cytoskeleton, the membrane skeleton so much, but also the membrane is sort of stabilized by a network of proteins that will define different regions within the membrane. This is the membrane skeleton here. You get a micrograph actually showing the membrane skeleton. Okay, here's an example of a lipid raft. And so we might have one part of the membrane here that has a particular composition, but then there's this other part of the membrane that is enriched in sphingolipids here, maybe has a little bit more cholesterol, and there'll be particular proteins associated just with these lipid raft regions within the membrane. Here I showed you this before. This is cholesterol within the membrane, and it basically helps aid the fluid. It makes it more stiff or less fluid. Okay, well, then we're going to get into membrane proteins. All right, so we've talked a little bit about the lipids with the membrane for the fact that they're so heterogeneous and sort of the different ways that they're heterogeneous. But let's talk about this component of the membranes that is actually the greatest component by weight, that is the proteins. When we talk about protein structure, we didn't talk much about membrane proteins, so here's our chance. There's three ways proteins can actually be associated with a membrane. One are integral membrane proteins. And an integral membrane protein is one that the whole protein spans across the membrane. And the secondary structural element that'll be associated with that membrane spanning area can be either transmembrane alpha helices, or transmembrane beta sheets. And some of these sheets will like wrap around and they form a thing known as beta barrels. I'll show you a picture of this in a bit. Okay, so that's where the protein actually sits within the membrane. The other way proteins can be associated with the membrane are lipid-linked proteins. And in a lipid-linked protein, an amino acid will be modified to actually add a little bit of a hydrophobic section, either a fatty acid to it or to a little isoprenoid tail. Okay, and so all of these, have a hydrophobic molecule covalently attached to the peptide backbone, and then that hydrophobic molecule embeds itself into the membrane. It's like an anchor, basically, kind of an anchor. And so what are these different hydrophobic molecules? There can be a small isoprenoid chain. Okay, and these are known as prenolated proteins. Usually 15 or 20 carbons long. So a C15, 15 little isoprene chain in there. It'll be attached, usually at the carboxy terminus, and then that anchors into the protein. That sort of anchoring really got a lot of study and became really about, the early 1990s or so, when it was discovered the RAS protein is prenolated protein. So some of the proteins involved in the signal transduction process that induces cell proliferation as part of cancer involves prenolation. A lot of this too is reversible. So basically a protein will become prenolated, it'll associate with a protein, and then it'll get cleaved off and kind of go back and forth. Anyway, so once people realized that protein prenolation was an important process in oncogenesis, then people started studying it like crazy. Or other than a small isoprenoid chain, the other way of looking at it is or add a fatty acid. These are known as acylated proteins. And the third way, I'm going to write this out one, is glycosyl phosphatidylinositol. Glycosyl phosphatidylinositol inositol linked proteins. This is conveniently known as GPI anchors. And And I'll show you a picture of the structure of that in a second. All right, so there's three different ways proteins are associated with the membrane. One is their integral membrane proteins. The other is these lipid links that basically provide anchors. And the other one is sort of secondary, is these are known as peripheral membrane proteins. And these are basically proteins that through electrostatic interactions, hydrogen bonding, salt bridges, and that sort of stuff, are associated with either an integral membrane protein or a lipid link protein. So these are linked to integral membrane proteins. Or lipid link proteins. Via electrostatic interactions.
Professor, are they normally on the inside of the cell or outside or both?
Both.
Okay.
And you also have to remember that we're talking about generically, so we're not just talking about the plasma membrane. So this can also be the the membranes of the Golgi apparatus, the membranes of the endoplasmic reticulum, or the nuclear membrane, or what have you. So let me try to show you some pictures that might illustrate some of these ideas a little bit more clearly. Okay, so here's an example of an integral membrane protein, rhodopsin. Okay, and what we have here is, in this light blue or whatever teal, is it what you call that, teal? I don't know. That's the phospholipid bilayer. And then in yellow we have our protein, and we see a whole bunch of transmembrane alpha helices. And there are multiple things. And you can actually take the primary sequence of a protein and ask it to predict whether or not there are any transmembrane domains. And it will come in and it will tell you whether or not there's a transmembrane, predicted to be a transmembrane domain. Because obviously in these alpha helices, what you would expect is more hydrophobic side chains in these alpha helices that will interact with the hydrophobic core of the phospholipid bilayer. Oh, they don't necessarily have to go all the way across the membrane. This is a cyclooxygenase. I don't know if you remember the cyclooxygenase. This is the enzyme that is inhibited by the non-steroidal anti-inflammatory drugs. It's the one that takes arachidonic acid as a substrate to make these inflammatory prostaglandins, that sort of thing. So it's actually a membrane protein, but what it does is it just have an alpha helix, some alpha helices that are hydrophobic that will embed in there without actually going all the way across the membrane. And this kind of makes sense because the substrate for the molecule, the arachidonic acid, is hydrophobic and it exists within the membrane. So the enzyme basically has to pick up that hydrophobic substrate, get it into in here, but then the product is actually soluble, and so the product then gets released out into the environment. Here's some examples of transmembrane beta barrels. So these are good. So the beta sheets that basically wrap around themselves. And what's kind of cool about these is then it creates a lumen inside the barrel. And that's actually, we're going to talk about transporters and carriers and that sort of thing. But that basically provides a space for stuff to get in and out across the membrane in a controlled fashion. So here's several different examples of those. Yeah, so tryptophan and tyrosine tend to cluster at the lipid-water interface. That's one more. Okay, then there's peripheral membrane proteins, which will be interacting with the transmembrane protein along that way. And these can be, so the way that membranes are studied is you generally isolate a membrane fraction, usually by centrifugation. And then, so you can isolate the membrane from all the other cellular components. Then you might take that isolated membrane, and it has the proteins and the lipids, the carbohydrates in there. But if you treat it with high salt, what you'll do is you'll interfere with these electrostatic interactions, and you'll release these peripheral membrane proteins. So essentially, there's an operational definition of peripheral membrane proteins, are the proteins that are released upon treatment of a membrane preparation with high salt. Whereas those ones that are more integral won't be released from the membrane. Okay, here's some examples, some anchors, okay? Acyl chain, so this is where we have a fatty acid that acts as an anchor. The fatty acid actually makes a thioester to a cysteine residue. Okay, that's how it's attached to the protein here, and that'll anchor here. It tends to be either C16 or C14 fatty acids that will act as the anchor. Then here's a similar idea, but now we have an isoprenoid side chain, in which this one example is 15 carbon, but also the 20 carbon version can inject itself in there. And then here's that GPI anchor, which is there. So we basically have, there's a inositol with a sugar, which is then attached to a phospholipid here. So you have the acyl chain of the phospholipid plus another acyl chain esterified to it. So they're basically three hydrophobic tails anchored. Then there's a series of sugars that are all linked together. Then it's eventually attached to the protein. So it's a little bit more complex structure. But basically the idea is that still it anchors it to the membrane. Okay, now we're getting into transport here. All right, let's go ahead and make it through. We're going to talk about membrane transport. I don't know if I'll make it through all of it, but we'll talk about most of it anyway. So now the question is, a membrane basically defines different regions, either the inside or outside of the cell, the inside or outside of the mitochondrion, the inside or outside of the nucleus. And one of the roles of the membrane is basically control the flux of materials and information between these different compartments. So the question, well, how does that actually happen? All right. So first of all, there's non-mediated transport. What do I mean by that? I mean, if there's some stuff that can get across the membrane without any help from any enzymes or anything like that. So what are some of the things that can get across the membrane? So this is, diffusion of gases and water. Oxygen Eventually, it has to make its way into the mitochondrial matrix, you know, next to the electron transport chain, right? So it has to diffuse across a bunch of membrane, be able to get there. The CO2 released has to diffuse out of the mitochondria and out of the cell, and those gases can just diffuse across. Water generally can also diffuse across a biological membrane, although there are certain cell types that, particularly in the kidney, that require very high rates of water flow, and those have special carriers we'll talk about called aquaporins. But generally, water can get in and out. That's why if you take an erythrocyte, a red blood cell, and you put it in a hypotonic solution, the water can flow in and make the cell bust, and you can generate the erythrocyte ghost. All right. But most of the transport is mediated transport. Now, what you can think about is it's easier to transport something if there's a pre-existing gradient. So if you have higher amount of glucose outside the cell than inside, energetically it's easier. You just got to control the flux. You just got to help carry it across the membrane, but there's no energetics available. Sometimes, though, you have to actually go against a concentration gradient, and that's where you have active transport. But what we'll first talk about is passive mediated transport. This is also known as facilitated diffusion. And you can have things like carriers. And a carrier, I guess in a kind of a diagrammatic way, you can think of a carrier like this. You can think about if here's your phospholipid bilayer. Maybe you have some sort of molecule, maybe some ion or something. I'll maybe just put something with a positive charge. It's going to come in here. Maybe that'll induce a conformational change. And it can go out. And as long as there's a concentration gradient there already, then you're just basically enabling it to sneak past.
Professor, is the membrane vertical in that picture?
Yes,
Okay.
yes.
Thank you.
Yeah. What's that? How that looks like. No, I guess what you need to know is the concept that a carrier is something that will basically bind a molecule and allow it to pass through the membrane, release it on the other side. That enables the movement along a concentration gradient, right? You're not trying to go, it's energetically favorable.
Okay.
So does that not require ATP?
What's that?
Does that not require ATP to do that?
That doesn't require ATP,
Okay.
yeah. But then we also have channels. And channels basically provide provide a space for the stuff to actually go through. And there are different flavors. We can talk about ion channels. Ion channels form pores. That allow diffusion of ions. We have what are known as porins. These are beta barrel proteins. That allow the selective passage of molecules, more than just ions. And I'll show you a picture in a little bit. You got the beta barrel that's going to go across the membrane, and so it basically provides a hole in the membrane. You think, well, anything can go through that hole, but what you'll have is specific amino acid residues that stick out from the beta barrel into the middle, and they'll be arranged stuff like that. Only certain molecules can actually make it through that beta barrel, so you get the selectivity of that way. And the next one is aquaporin, so it's another type of channel. And I'm going to show you a picture of these. They're particularly cool because these allow for the passage of water, but not solutes. Actually, I didn't, I'm going to come back. We're going to, what we're going to do is we're going to go ahead and stop there. What I forgot to do is I load up a few of the slides to get to this channel. So we're going to go ahead and stop there. And then when we come back, I'll illustrate those with some examples of different channels and porins and that sort of stuff. Then we'll talk briefly about active transport. That might take 10 or 15 minutes or so. Then we're going to jump into the next section where we get the metabolism. We're going to start moving away from the survey of biological molecules and start talking about how they interact on Monday. Remember, no class on Wednesday. I'll figure out how to get through all of this.